Good morning, everyone. I'm glad to see you here today. So I would like to present you our work with the title of LIBS technique applied for high precision analysis of transition metals and alkaline pollutants in human physiological matrices. So generally speaking, like background knowledge, in, for example, biomedical field or pharmaceutical field or even environmental studies, we need to have some kind of tool or instrument that we could use to have to be able to find some contamination and pollutants in very low concentration, because it's like it's a question of our health, of our environment, and our well-being. So we have different kinds of techniques that are going to pursue these kinds of goals. For example, here I say something about the spectroscopic techniques like Raman, infrared, and fluorescence. These techniques are well known in the biomedical field, but these techniques are molecular techniques, so they study the, for example, rotational, vibrational energies of different kinds of compounds. So they are great for organical compounds, but for example, if you want to study the ion contents, it's really important to have some kind of tool that can make it in life. So in our case, we chose LIBS technique, because LIBS technique, or laser-induced breakdown spectroscopy, is atomic emission spectroscopical technique that uses high energy pulses to create a plasma. So here, for example, is like a standard LIBS experiment, the basics. So here are the few steps you need to make to achieve the correct spectra after the experimental setup. So here we produce the high pulse energy with the ND YAG laser that operates at 1064 nanometers. After that, the laser pulse is collected by the optical system, and after that we focus it in specific spot in the sample, and we create the plasma. So plasma is basically the fourth state of matter. It means that we, with this high energy, the voltage, the heat, we excite the molecules within the sample, and we create some kind of excitations, after which we collect the photons that were excited via optical fiber, and after that we create the spectra. So spectra here are, these are the emission lines from atoms that are in the sample. So the important part of this work is sample preparation. We chose the Higgs-based salt solution to create a chemical environment that mimics the composition and the condition of a physiological matrix. So, for example, buffer system, the phosphate and the carbonate buffers. So here you see how we created it in a few simple steps. We chose the salts. I will talk about them later. These salts we were adding in the water and heating it at 37 degrees of Celsius. So we created 100 milliliters of it. After that, we stored it in a fridge for +4 degrees of Celsius, and we stored it for 1 week to see if there is going to happen any precipitation, because if there is a precipitation, it means the buffer system are not working well. So that's why we need so much time to spend at this point. And after the sample preparation, we took our Higgs-based salt solution, add the contaminants such as chromium oxide and also copper chloride. And after that, we see the given concentration. So we had basically 4 of them. So for copper it was 0.79 millimolars, 0.316, 0.158, and also 0.079. So this is like the goal of ours to reach, to be able to found in these kind of solutions. So here you see the ion content. So after we created the sample, we took our information and calculated the ion concentration in the sample, and also we compared it to the human blood plasma. So here you see it's almost reachable, but we had some other concentrations in that cases for the buffers. And also these are the chemical compounds that we use to create this physiological matrix. So What I will return to LIBS in liquids because there are some specifications. LIBS is great for solids because you can just study the alloys. But in liquids, there are a few advantages and disadvantages. I would like to start with the advantages. So there is no sample preparation. If you want to study a sample, for example, in ICP-MS, it's the most high precision technique we have in our days. You need to add the nitric acid to make a combustion, and it can damage or make a different ion content of a sample. So we didn't want it. So we used the LIBS because basically you just take a sample and make shot with the laser and you're done. So also the remote control and sensitivity is important part in LIBS because remote control doesn't make it more contaminants with how we manipulate the sample, and sensitivity is how we can reach this goal that we put to ourselves. But there are a few challenges. So water interference and quenching and splashing. So water interference because of the density of the molecules of water. It can be possible that water just wants to eat the laser balls. So we just don't get the proper analyzation after that. So we address this problem with the experimental setup. We use the acoustical levitations. So here you see the droplet. We made it with the needle and also the IR laser. Infrared laser was used to heat, to dry the droplet. And also the preconcentration factor is the initial volume over the final volume was rated at like 50. So the volume of the droplet was approximately 20 microliters. So after that, we created the droplet, shoot with the laser, we are done. So this is the video, how we can create it. So in our lab, we have a needle automatically splashing or making the droplet right now. But this is how you, in principle, make the droplet. So it just stays and does whatever it wants. So finally, the results. So this is the spectra we got from the HBSS solution without any contaminants. So here you see the sodium doublet. This is the highest line we got because sodium is it's by nature like that. And also the potassium, the nitrogen, chlorides, magnesium, calcium. These are all that we got. These are all the elements we got from the compounds. So it was important to us to see if we could see these elements without interference in them. And after, it was important to us to study the contaminations. So the copper spectra, for example, here you see two lines at 324 and 327 nanometers, and we also compared it to the blanks. So you see a very high intensity. So this is intensity, this is the wavelength. And the black line is the copper. And we also calculated the LODs. LODs for copper was 0.0997. So, and also we did the same for chromium. Here you see the same situation about the chromium, but also we can see the calcium and also the calcium another line. So these lines are not interfering, so we can properly characterize them. So LOD was at this point 0.099, and I would like to start a discussion about it. So the minimal concentration of copper cannot be quantified with the 95% confidential rate because the LOD value was too big to analyze the minimal concentration we prepared. And also, as the result, we can conclude that for this kind of analysis, chromium has shown more sensitivity than copper due to the spectral response from the chromium. However, in both cases, we can say that LIBS and acoustical levitation is reliable, can be used to make a reliable quantification in millimolars order. So this is the end of our study. And also for future plans, this study is preliminary because it's a part of my bachelor thesis. So after that, we want to prepare and compare results from LIBS, AL, and SEN-LIBS. So we will create the surface enhanced LIBS and compare what are what is the best technique for that. I would like to thank the VPS projects and all the projects that are listed here, and also my Dr. Ivan Alexander Medina, yes, and also Pavel Weiss for being an important part in this work. And so thank you for your attention. Yes.